Basics of Medical Biochemistry. First edition, 2022. Chapter 10. Metabolism of Carbohydrates, Part 3 of 3. By Professor Dr. Muhammad Jawad. MBBS, Dow, M. Philosophy, PhD, Dip M.E. Head of the Biochemistry Department Faiz-i Ruthfa Medical College PAF Base Faisal Karachi Pakistan. Ex-Professor of Biochemistry, Qasim University Kingdom of Saudi Arabia. Ex-Professor, Head of the Biochemistry Department and Vice Principal. Liaquat College of Medicine and Dentistry and Fatima Jinnah Dental College, Karachi, Pakistan. 10.12 Gluconeogenesis. It is the formation of new glucose, i.e., the formation of glucose from non-carbohydrate sources. Gluconeogenesis fulfills the needs of the body for glucose when sufficient carbohydrates are not available from the diet or glycogen reserves. A supply of glucose is necessary, especially for the nervous system and erythrocytes. Failure of gluconeogenesis is usually fatal. The liver and kidneys are the main organs to perform gluconeogenesis. They contain all enzymes required for gluconeogenesis. Gluconeogenesis occurs intracellularly, mainly in the cytosol, and only one reaction, carboxylation of pyruvate, takes place in mitochondria that need pyruvate carboxylase enzyme. 90% of gluconeogenesis takes place in the liver and 10% in the kidney. The liver can produce 1 kg of glucose per day. The following are the sources of new glucose: pyruvate and lactate, muscle, RBC. Amino acids, skeletal muscle. Glycerol, fat of adipose tissue. 10.12.1 Gluconeogenesis from pyruvate. Gluconeogenesis using pyruvate is essentially the reverse of glycolysis, except for the 3 irreversible reactions that need different enzymes for the reversal. These irreversible reactions are pyruvate to phosphenol pyruvate, PEP, fructose 1,6-bisphosphate to fructose 6-phosphate, and glucose 6-phosphate to glucose. 1 Conversion of pyruvate to phosphenol pyruvate. First bypass of irreversible glycolytic step. This takes place in 2 steps. The first, pyruvate is converted to oxalostate, which is then changed into phosphenol pyruvate. The conversion of pyruvate to oxalostate takes place by the enzyme pyruvate carboxylase in the presence of CoO2 and ADP. Biodin binds with CoO2 from bicarbonate as carboxybiodin before the addition of CoO2 to pyruvate. Pyruvate plus CoO2 plus ADP, oxalostate plus ADP plus pi. Pyruvate carboxylase is a biodin-dependent mitochondrial enzyme, whereas the other enzymes of gluconeogenesis are cytoplasmic. Excessive consumption of raw eggs can cause nutritional deficiency of biodin. As avidin, a protein found in egg whites, can tightly bind with biodin. Transport of oxalostate to the cytosol. Oxalostate is synthesized in the mitochondrial matrix. It has to be transported to the cytosol to be used in gluconeogenesis. Malate shuttle. Mitochondrial membrane is impermeable to oxalostate, while permeable to malate. So, oxalostate is reduced to malate inside the mitochondria for shifting to the cytosol. The reduction is accomplished by NADH-linked malate dehydrogenase. When malate has been transported across the mitochondrial membrane, it is reoxidized to oxalostate by an NAD+ linked malate dehydrogenase in the cytosol. This enzyme is present in both mitochondria and cytosol. Figure 10.22 Malate shuttle, IMM, inner mitochondrial membrane. In the second step, oxalostate is converted to phosphenol pyruvate through decarboxylation and phosphorylation. This reaction is catalyzed by phosphenol pyruvate carboxygenase, using GTP as the phosphate donor. Oxalostate plus GTP, phosphenol pyruvate plus GDP plus CoO2. The biological significance of decarboxylation of oxalostate. In the liver and kidney, the reaction of succinate thiokinase in the TCA cycle produces GTP, rather than ADP as in other tissues, and this GTP is used for the reaction of phosphenol pyruvate carboxygenase. Thus, providing a link between TCA cycle activity and gluconeogenesis. To prevent excessive removal of oxalostate for gluconeogenesis, which would impair TCA cycle activity. Phosphenol pyruvate now follows the route of glycolysis simply in the reverse direction until the next irreversible step is reached, which is the formation of fructose 1,6-bisphosphate. 2. Conversion of fructose 1,6-bisphosphate to fructose 6-phosphate. Second bypass of irreversible glycolytic step. The metabolism of phosphenol pyruvate by the enzymes of glycolysis in the reverse direction is near equilibrium under intracellular conditions, so when conditions favor gluconeogenesis, the reverse reactions will take place. This hydrolysis of fructose 1,6-bisphosphate to fructose 6-phosphate and pi is catalyzed by the enzyme fructose 1,6-bisphosphatase. This enzyme is present in the liver, kidney, and skeletal muscle, but is perhaps absent in the heart and smooth muscle. It is an allosteric enzyme and participates in the regulation of gluconeogenesis. Fructose 1,6-bisphosphate plus H2O fructose 6-phosphate plus pi. The fructose 6-phosphate is readily converted into glucose 6-phosphate by phosphohexose isomerase or phosphoglucose isomerase. Fructose 6-phosphate glucose 6-phosphate. This is the end of gluconeogenesis in most tissues. Free glucose is not generated, rather, the glucose 6-phosphate is utilized in some other manner, particularly to form glycogen. One advantage to ending gluconeogenesis at glucose 6-phosphate is that, unlike free glucose, it cannot diffuse out of the cell. 3. Conversion of glucose 6-phosphate to glucose. Third bypass of irreversible glycolytic step. The enzyme glucose 6-phosphatase catalyzes the conversion of glucose 6-phosphate to glucose. An associated Ca2+ binding stabilizing protein is essential for phosphatase activity. Glucose 6-phosphate plus H2O glucose plus pi. This final step in the generation of glucose does not take place in the cytosol. Rather, glucose 6-phosphate is transported into the lumen of the endoplasmic reticulum, where it is hydrolyzed to glucose by glucose 6-phosphatase, which is bound to the membrane. Glucose and pi are then shuttled back to the cytosol by a pair of transporters. 10.12.2 Energetics of gluconeogenesis from pyruvate. During the synthesis of glucose from pyruvate in gluconeogenesis, 6 molecules of nucleotide triphosphate, 4 ADP and 2 GTP, are hydrolyzed. Whereas only 2 molecules of ADP are generated in glycolysis in the conversion of glucose into pyruvate. Thus, it is not a simple reversal of glycolysis, but it is energetically an expensive business. The overall reaction of glycolysis is glucose plus 2 NAD plus 2 ADP plus 2 pi 2 pyruvate plus 2 NADH plus H plus 2 ADP plus 2 H2O. The overall reaction of gluconeogenesis, for conversion of 2 pyruvates to 1 glucose, is 2 pyruvate plus 4 ADP plus 2 GTP plus 2 NADH plus H plus 4H plus 4 H2O glucose plus 4 ADP plus 2 GDP plus 2 NAD plus 6 pi. Figure 10.23 Reactions of gluconeogenesis from pyruvate to glucose. 10.12.3 Gluconeogenesis from lactate. The formation of glucose from lactate takes place via the Cori cycle. Lactate is generally produced by anaerobic glycolysis in the muscles during exertion. Lactate is then released from the muscles to the blood and travels to the liver for conversion to pyruvate and, ultimately to glucose. The biological significance of gluconeogenesis from lactate. During anaerobic glycolysis in skeletal muscle, pyruvate is reduced to lactate by lactate dehydrogenase, LDH. This reaction serves 2 critical functions during anaerobic glycolysis. First, in the direction of lactate formation, the LDH reaction requires NADH and yields NAD+, which is then available for use by the glyceraldehyde 3-phosphate dehydrogenase reaction of glycolysis. These 2 reactions are, therefore, closely coupled during anaerobic glycolysis. Secondly, the lactate produced by the LDH reaction is released into the bloodstream and transported to the liver, where it is converted to glucose. The glucose is then returned to the blood for use by muscle as an energy source and to replenish glycogen stores, Cori cycle. 10.12.4 Gluconeogenesis from amino acids, all amino acids except lysine and leucine, derived from dietary proteins, tissue proteins, or from the breakdown of skeletal muscle proteins during starvation can give glucose. The carbon skeletons of these amino acids yield either pyruvate or intermediates of the citric acid cycle after transamination or deamination. The most important glucogenic amino acid is alanine. Glucosalanine cycle. Alanine is formed in peripheral tissues, muscle, by the transamination of pyruvate. The pyruvate is formed by aerobic glycolysis as well as from the catabolism of branched-chain amino acids, isoleucine and valine. In the liver, alanine is transaminated with ketoglutarate by ALT to give pyruvate and glutamate. Pyruvate is converted to glucose, gluconeogenesis, and transported to muscles. Glutamate is deaminated by glutamate dehydrogenase to produce ammonia. Figure 10.24 Glucosalanine cycle. Source: Online Biochemistry Notes. Entry of glucogenic amino acids to pyruvate and intermediates of the TCA cycle. Pyruvate is the point of entry for alanine, cysteine, glycine, serine, threonine, and tryptophan star. Oxalostate is the point of entry for aspartate and asparagine. Aspartate, a 4-carbon amino acid, is directly transaminated to oxalostate. Alpha-ketoglutarate is the point of entry for several 5-carbon amino acids, arginine, glutamate, glutamine, histidine, and proline, that are first converted into glutamate. Succinyl-CoA is a point of entry for some of the carbon atoms of isoleucine *, methionine, and valine. Propionyl-CoA and then methylmalonyl-CoA are intermediates in the breakdown of these 3 nonpolar amino acids. Threonine is also converted to propionyl-CoA. Fumarate is the point of entry for aspartate, phenylalanine * and tyrosine *. These amino acids are also ketogenic. Figure 10.25 Entry of glucogenic amino acids to intermediates of TCA cycle. 10.12.5 Gluconeogenesis from propionate. In humans, propionate arises from the beta-oxidation of odd-chain fatty acids, as well as the oxidation of isoleucine and the side chain of cholesterol. Figure 10.26 Formation of succinyl-CoA from propionate. Propionate is a major precursor of glucose in ruminants. It enters gluconeogenesis via the TCA cycle. After esterification with CoA, propionyl-CoA is carboxylated to dimethylmalonyl-CoA, catalyzed by propionyl-CoA carboxylase, a biodin-dependent enzyme. Methylmalonyl-CoA racemase catalyzes the conversion of dimethylmalonyl-CoA to L-methylmalonyl-CoA, which then undergoes isomerization to succinyl-CoA catalyzed by methylmalonyl-CoA mutase. Methylmalonyl-CoA isomerase/mutase is a vitamin B12 dependent enzyme. That's why in vitamin B12 deficiency methylmalonic acid is excreted in the urine. 10.12.6 Gluconeogenesis from glycerol. Glycerol and fatty acids are produced as a result of the hydrolysis of triacylglycerols in fat cells. Glycerol may enter either the gluconeogenic or the glycolytic pathway after converting to dihydroxyacetone phosphate. Glycerol released from the lipolysis of triacylglycerols in adipose tissue is used solely as a substrate for gluconeogenesis in the liver and kidneys, in the fasting state. This requires the phosphorylation of glycerol to glycerol 3-phosphate by glycerol kinase and dehydrogenation to dihydroxyacetone phosphate by glyceraldehyde 3-phosphate dehydrogenase. Figure 10.27 Formation of dihydroxyacetone phosphate from glycerol. Glycerol released from hydrolysis of triacylglycerols cannot be utilized for reesterification, as glycerol kinase is absent in adipose tissue. It is transported through the circulation to the liver and used for gluconeogenesis or glycolysis as per the need of the body. 10.12.7 Significance of gluconeogenesis. Glucose is a very key substance for metabolism and its continuous supply is essential to the body for a variety of functions. Liver glycogen can meet the body's needs for glucose only for 10 to 18 hours without dietary carbohydrates. Gluconeogenesis supplies body cells with glucose after 4 to 6 hours of the last meal and becomes fully active as stores of glycogen are depleted and continuously add glucose to the blood. Production of glucose by gluconeogenesis is necessary to maintain the required glucose levels in the blood. As the brain, erythrocytes and muscles in exertion use glucose at a rapid rate and require glucose between meals, especially after several hours. The human brain alone requires about 120 g of glucose per day, out of the 160 g needed by the entire body. Glucose is the only source that supplies energy to the skeletal muscles under anaerobic conditions. The degradation of glycogen in muscle results in the formation of lactate. The breakdown of fat in adipose tissue produces free glycerol and propionate. Gluconeogenesis helps in clearing these metabolites, which accumulate in the blood. Lactate, glycerol, propionate, and some amino acids are good precursors for glucose synthesis. The Cori cycle is responsible for the conversion of muscle lactate to glucose in the liver. 10.12.8 Regulation of gluconeogenesis. Gluconeogenesis and glycolysis are coordinated within a cell in such a way that one pathway is relatively slow while the other is quite rapid. The amounts and activities of the specific enzymes of each pathway are controlled to keep a required balance among them. The main regulators of gluconeogenesis are glucagon and the availability of glucogenic substrates. Gluconeogenesis and glycolysis are regulated by similar effector molecules but in opposite directions. Low blood sugar results in high gluconeogenesis and low glycolysis. High blood sugar results in low gluconeogenesis and high glycolysis. Role of glucagon in the regulation of gluconeogenesis. Glucagon is a peptide hormone synthesized by alpha cells of islets of Langerhans of the pancreas. It is catabolic in action, as it mobilizes glucose, fatty acids, and amino acids from their respective stores into the bloodstream. It increases plasma glucose by stimulating hepatic glycogenolysis and gluconeogenesis. It stimulates gluconeogenesis by following three mechanisms. 1. Glucagon activates the fructose 1,6-bisphosphate by lowering the hepatic fructose 2,6-bisphosphate. At the same time, PFK1 is inhibited and thus situation becomes favorable for gluconeogenesis. 2. Glucagon increases the concentration of CAMP and CAMP-dependent protein kinase, leading to the phosphorylation and inactivation of pyruvate kinase. As a result, the conversion of phosphenol pyruvate to pyruvate is decreased and phosphenol pyruvate is diverted toward gluconeogenesis. 3. Glucagon increases the synthesis of phosphenol pyruvate carboxygenase by increasing its transcription gene. Cortisol also has the same effect but insulin has the opposite effect. Role of substrate availability in the regulation of gluconeogenesis. Changes in the availability of substrates are responsible for most changes in the rate of gluconeogenesis. Low insulin levels facilitate the release of glucogenic amino acids and promote gluconeogenesis. ADP and NADH are also the requirements of gluconeogenesis. These are supplied by the oxidation of fatty acids. Role of acetyl-CoA in the regulation of gluconeogenesis. Acetyl-CoA is an allosteric activator of pyruvate carboxylase. It is derived from the lipolysis of triacylglycerols in adipose tissue and the subsequent oxidation of fatty acids. The activation of pyruvate carboxylase and the reciprocal inhibition of pyruvate dehydrogenase complex by acetyl-CoA explain the action of fatty acid oxidation in sparing the oxidation of pyruvate and in stimulating gluconeogenesis. Role of AMP in the regulation of gluconeogenesis. A high level of AMP indicates that the energy charge is low and signals the need for ADP generation. On the other hand, high levels of ADP and citrate indicate that the energy pool is high and that biosynthetic intermediates are abundant. Under these conditions, glycolysis is nearly stopped and gluconeogenesis is stimulated. Phosphofructokinase 1, the chief regulatory enzyme of glycolysis, is also subject to feedback control. It is stimulated by AMP but inhibited by ADP and citrate. Fructose 1,6-bisphosphatase, enzyme for the second bypass of the irreversible glycolytic step, is inhibited by AMP and activated by citrate. Figure 10.28 An overview of the regulation of gluconeogenesis. Reciprocal regulation of gluconeogenesis and glycolysis in the liver. Glycolysis and gluconeogenesis are reciprocally regulated. When glycolysis is on, gluconeogenesis is turned off, especially in the well-fed state, whereas under conditions of starvation, gluconeogenesis is fully active and glycolysis is turned off. Both cycles are never active at the same pace at the same time. Fructose 2,6-bisphosphate allosterically activates the phosphofructokinase 1, the enzyme of glycolysis. Fructose 2,6-bisphosphate also activates the transcription of the gene for glucokinase, which phosphorylates glucose to glucose 6-phosphate at the start of glycolysis. Phosphofructokinase 1, the rate-limiting step of glycolysis, is allosterically inhibited by ADP. Figure 10.29 Reciprocal regulation of gluconeogenesis and glycolysis in the liver, F, fructose, BP, bisphosphate. 10.13 Metabolism of ethyl alcohol, ethanol. Alcohol is consumed in considerable amounts by many people. About 20% of ingested alcohol is absorbed in the stomach. It is metabolized by the liver in 2 steps, leading to the formation of acetyl-CoA. 1-ethanol is first oxidized into acetaldehyde by alcohol dehydrogenase, ADH, which is located in the cytoplasm. CH3CH2O plus NAD plus CH3CHO plus NADH plus H plus. 2-acetaldehyde is further oxidized into active acetate, acetyl-CoA, by aldehyde dehydrogenase, ALDH, predominantly associated with mitochondria. O. 2. CH3CHO plus COH plus NAD plus CH3CS-CoA plus NADH plus H plus. Acetyl-CoA is oxidized in the TCA cycle, converted to fatty acids, cholesterol, or utilized in an acetylation reaction. Effect of increased NADH/NAD plus. Inhibition of fatty acid oxidation, increased fatty acid esterification producing more triacylglycerol resulting in a fatty liver. Decreasing the activity of the TCA cycle. Increasing lactate/pyruvate ratio, resulting in hyperlactic acidemia. In this way, alcohol may contribute to obesity. Inhibition of gluconeogenesis from lactate. In addition, acetaldehyde is more toxic than ethanol. It is also metabolized to a free radical intermediate, 1-hydroxyethyl radical, which initiates free radical reactions in lipids and other molecules. Alcohol also appears to increase metabolic rate significantly, thus causing more calories to be burned rather than stored in the body as fat. In chronic alcoholism or high consumption of alcohols, ethanol metabolism takes place via a cytochrome P450 enzyme involving NADPH and O2 in the endoplasmic reticulum. The hydrogen released by this reaction is bound to oxygen and to NADPH to form water and NADP+. This reaction takes energy rather than producing it. Ethanol can inhibit the metabolism of some drugs by competing with cytochrome P450 dependent enzymes. 10.14 Regulation of blood glucose levels. The blood glucose level is mainly regulated by the entry of glucose into the bloodstream and its exit for utilization elsewhere in the body. Normal fasting blood glucose level, 8 to 12 hours after carbohydrate meal, is 70 to 110 mg/dl. In a healthy individual, it arises to 120-150 mg/dl 1 hour after a meal but returns to a normal fasting level after 2 hours, 2 hours postprandial. The level of blood glucose is maintained within the normal range by the action of 2 opposing factors. 1. The rate of glucose entrance to the blood. Glycogen breakdown and gluconeogenesis. Dietary carbohydrates. 2. The rate of removal of blood glucose. Production of energy by all tissues. Glycogen synthesis. Lipogenesis. Hyperglycemia occurs when the blood glucose level rises more than 180 mg/dl. It is mainly due to a deficiency of insulin that leads to decreased glucose utilization by the body tissues. Other causes of hyperglycemia include an increased rate of gluconeogenesis, decreased rate of glycogenesis and decreased rate of glucose transport to certain tissues. Hypoglycemia occurs when the blood glucose level decreases to less than the normal range. Under normal circumstances, the blood glucose level is so efficiently regulated that it rarely rises above 160 mg/dl irrespective of high carbohydrate intake. Similarly, it seldom falls below 40 mg/dl even after starvation for a few weeks. Hyperglycemia is harmful in the long run, while hypoglycemia even for a short period is dangerous and may even be fatal. Figure 10.30 An overview of the regulation of blood glucose level. Source: Pearson Education. Benjamin Cummings, 2005. Hyperglycemic endocrine factors. Many hormones cause a rise in blood glucose levels. 1. Glucagon. It is the main hormone to maintain blood glucose levels during fasting. It causes glycogenolysis in the liver by the activation of glycogen phosphorylase, inactivation of glycogen synthase, and decreases the glycolysis. Glucagon increases the activity of phosphenol pyruvate carboxygenase, which stimulates gluconeogenesis. 2. Glucocorticoids. Cortisol. Glucocorticoids increase the blood glucose level by stimulating gluconeogenesis in the liver. They create hyperglycemia that may lead to adrenal diabetes. There is rapid gluconeogenesis from pyruvate and amino acids. They also reduce the uptake of glucose by cells as well as decrease glucose utilization. 3. Growth hormone. The effect of growth hormone to create hyperglycemia is twofold. Glucose uptake by muscle and adipocytes is decreased while glucose synthesis, gluconeogenesis, by the liver is stimulated. Along with hyperglycemia, glucosuria, and a diabetic-like tolerance curve are also produced. It also potentiates the hyperglycemic effects of glucagon and epinephrine. 4. Adrenocorticotropic hormone, ACTH. It stimulates the growth of the adrenal cortex and stimulates it to secrete glucocorticoids, cortisol. The cortisol in turn causes hyperglycemia. 5. Thyroid hormone. It increases intestinal absorption of glucose. Thyroid hormones stimulate almost all aspects of carbohydrate metabolism, including enhancement of insulin-dependent entry of glucose into cells and increased gluconeogenesis and glycogenolysis to generate free glucose. The net metabolic effect is hyperglycemia. 6. Catecholamines. These are the hormones of the adrenal medulla. Epinephrine and norepinephrine both cause glycogenolysis in the liver and skeletal muscles that leads to an increase in blood glucose level. Pheochromocytoma, a tumor of the adrenal medulla, overproduces catecholamines and is usually associated with hyperglycemia. 7. Antidiuretic hormone, ADH, or vasopressin. It is the secretion of the posterior pituitary gland or neurohypophysis. It causes an increase in the secretion of ACTH, which then causes an increased release of cortisol via excessive ACTH release. The cortisol in turn causes hyperglycemia by the direct effect of ADH on hepatic glycogenolysis through V1A receptors. 8. Somatostatin. It is secreted by the delta cells of islets of Langerhans of the pancreas. Although it inhibits the secretion of pancreatic hormones, including glucagon and insulin. But too much somatostatin results in an extreme reduction in the secretion of many endocrine hormones, like suppression of insulin leading to raised blood glucose levels. 9. Female sex hormones. The primary female sex hormones are estrogens, estrone and estradiol, and progesterone produced by the ovaries. They produce diabetic-like glucose tolerance in normal individuals. Moreover, the administration of estrogen to diabetic women aggravates their already existing diabetes. During pregnancy, progesterone increases insulin resistance in association with the production of the other placental counterregulatory hormones, including placental growth hormone, placental lactogens, placental corticotropin-releasing hormone and cortisol. Hypoglycemic endocrine factors. Some endocrine secretions produce hypoglycemia. 1. Insulin. Insulin is well-known hypoglycemic hormone. It stimulates glycogen synthesis via the stimulation of the enzyme glycogen synthase and inhibition of glycogen phosphorylase. The rate of glycolysis and HMP shunt is also increased by insulin. Some, 10%, hypoglycemic activity of the insulin is due to proinsulin. 2. Somatomedin or insulin-like growth factor, IGF. It is a protein produced in the liver and muscles. Its production is stimulated by growth hormone. Its primary function is to stimulate growth but also possesses some ability to decrease blood glucose levels. 3. Enteroglucagons or gut glucagons. These are glucagon-like peptides, GLP-1, secreted by intestinal cells in response to meals. GLP-1 stimulates insulin production by the beta cells. It reduces the postprandial rise in glucagon after meals, and both of those result in glucose lowering. Non-endocrine factors involved in the regulation of blood glucose level. 1. Gastrointestinal tract. The type of ingested carbohydrates has a direct impact on the postprandial blood glucose level. Complex carbohydrates are slowly digested and do not cause an abrupt rise in blood glucose levels. Whereas, hyperglycemia, called alimentary hyperglycemia, may occur after the intake of high amounts of simple sugars. Following the digestion of nutrients by the digestive tract, glucose is absorbed by the intestinal epithelium and then released in the blood flow of the portal vein to the liver. The liver is perfect enough to regulate postprandial glucose levels through glucose sensing mechanisms. Clinical aspects. There are some drugs, acarbose and miglitol, devised to reduce glucose absorption by inhibiting the disaccharidases in the alimentary canal. 2. Nervous system. It plays a partial role in the regulation of blood glucose levels. The autonomic division of the nervous system modifies the release of insulin and glucagon. The sympathetic stimulation that occurs with exercise stimulates glucagon formation, and this sustains blood glucose levels that would otherwise fall as muscles consume glucose for their energy. 3. Liver. The liver is the major organ that stores and supplies glucose for maintaining blood glucose levels. In response to hypoglycemia, the liver starts glycogenolysis to release glucose in the blood. The glycogenolytic effect of hypoglycemia occurs via sympathetic stimulation to increase the secretion of catecholamines. Secretion of growth hormone and glucagon is also increased in hypoglycemia. Both of these hormones are potent hyperglycemic agents. In response to hyperglycemia, insulin secretion is released more, which enhances the process of glycogenesis and gluconeogenesis in the liver. 4. Physical activity. Exercise causes increased glucose uptake from the blood into the muscle by activation of GLUT4. During exercise, blood glucose levels can be maintained or increased by increased release of glucose from the liver and kidneys into blood, as well as by the mobilization of other alternative fuels. Exercise stimulates glucagon secretion. In exhaustive exercise, the blood concentration of glucagon often increases fourfold to fivefold. A beneficial effect of glucagon is to prevent a decrease in blood glucose levels. One of the factors that might increase glucagon secretion in exercise is increased circulating amino acids. Other factors such as beta-adrenergic stimulation of the islets of Langerhans may also play a role in the secretion of glucagon. 5. Hypoglycemic drugs. Oral hypoglycemic drugs such as sulfonylureas, metformin, thiazolidinediones and alpha-glucosidase inhibitors are used frequently to control hyperglycemia. They are commonly used in the treatment of diabetes mellitus to lower glucose levels in the blood. Dipeptidyl peptidase 4, DPP4, inhibitors, sitagliptin, saxagliptin, linagliptin, and alogliptin, are a class of medicine that lower high blood glucose levels and may be used in the treatment of type 2 diabetes. DPP4 inhibitors inhibit the enzyme dipeptidyl peptidase 4 and prolong the action of glucagon-like peptides GLP-1. This inhibits glucagon release, increases insulin secretion and decreases gastric emptying, thus decreasing blood glucose levels. Table 10.7 Effects of hormones to regulate blood glucose level. Insulin. Glucagon. Lowers blood glucose. Promotes glycogen synthesis. Promotes glycolysis. Inhibits gluconeogenesis. Increases blood glucose. Promotes glycogenolysis. Enhances gluconeogenesis. Inhibits glycogen synthesis. Inhibits glycolysis. Cortisol. Epinephrine. Increases blood glucose level. Increases gluconeogenesis. Releases amino acids from the muscle. Increases blood glucose level. Promotes glycogenolysis. Increases gluconeogenesis. Favors uptake of amino acids. Growth hormone. Thyroid hormone. Increases blood glucose level. Decreases glycolysis. Mobilizes fatty acids from adipose tissue. Increases intestinal absorption of glucose. Increases gluconeogenesis. Increases glycogenolysis. 10.15 Hypoglycemia. Hypoglycemia occurs when the blood glucose level decreases to less than 50 mg/dl. Hypoglycemia can be classified into 1. Conditions in which there is a defect in the release of glucose by the liver, gluconeogenesis. 2. Conditions in which tissue glucose uptake is increased. It is, however, more convenient to distinguish hypoglycemia, which occurs as a response to fasting for 12 to 16 hours. Hypoglycemia due to some other stimuli, frequently called reactive hypoglycemia. Causes of fasting hypoglycemia. Insulinoma. Liver diseases. Hypoadrenalism, Addison's disease. Hypopituitarism. Glycogen storage diseases. Idiopathic hypoglycemia of childhood. Causes of reactive hypoglycemia. Drug-induced, usually due to insulin or oral hypoglycemic agents, example sulfonylurea, and also to dietary constituents, example alcohol and L-leucine. Galactosemia. Hereditary fructose intolerance. Essential reactive hypoglycemia, in which symptoms usually occur 2 to 4 hours after a meal, is probably due to an exaggeration of the normal insulin response to carbohydrate ingestion. Laboratory evaluation of hypoglycemia. The blood tests that help to diagnose the condition of hypoglycemia are the estimation of blood glucose, insulin, C-peptide, and proinsulin. The normal level of plasma insulin is approximately 42 to 243 pmol/L. The diagnosis of an insulinoma is usually made biochemically with low blood glucose, elevated insulin, proinsulin, and C-peptide levels. The serum concentration of C-peptide reflects the production of endogenous insulin by the pancreas and is not affected by insulin antibody interference. The normal serum level of C-peptide is 0.8 to 4.0 ng/ml. 10.16 Neonatal hypoglycemia. Neonatal hypoglycemia occurs when the blood glucose level is less than 30 mg/dl, 1.65 mol/L, in the first 24 hours of life and less than 45 mg/dl, 2.5 mol/L, thereafter. Neonatal hypoglycemia is the most common metabolic problem in newborns. The overall incidence is 5/1000 live births. Normal newborns may be hypoglycemic in 10% of cases if feeding is delayed for 3 to 6 hours after birth. 30% of normal infants are still at risk of hypoglycemia due to other reasons. Why is neonatal hypoglycemia more serious than in adults? Glucose is the primary fuel for the brain. The brain needs a steady supply of glucose to function normally. Glucose is the only source of carbohydrates for the fetus. Compared with adults, infants have a higher brain to the body weight ratio, resulting in higher glucose demand with glucose production capacity. Cerebral glucose utilization accounts for 90% of the neonate's glucose consumption. Fetal plasma glucose is 60 to 80% of the maternal glucose level. The fetus stores glucose in the form of glycogen, liver, heart, lung, and skeletal muscle. Most of the glycogen is made and stored in the last month of the third trimester. The fetus has a limited ability to convert glycogen to glucose and must rely upon the placental transfer of glucose to meet energy needs. When the infant is born, the cord is cut. As a result, the major supply of glucose is lost. The transition from fetus to newborn creates a significant energy drain on the newborn. The newborn is now required to meet increased metabolic demands while changing the energy source from a placenta to an external food source. Table 10.8 Risk factors and clinical features of neonatal hypoglycemia. Risk factors. Sign and symptoms. Gestation less than 37 weeks. Infant of a diabetic mother. Small for gestational age. Large for gestational age. Stressed/ill infants. Exposure to certain medications. Treatment of preterm labor. Treatment of hypertension. Treatment of type 2 diabetes. Benzothiazide diuretics. Tricyclic antidepressants in the third trimester. Jitteriness, nervousness. Irritability. Hypotonia. Lethargy. High-pitched cry. Hypothermia. Poor suck. Tachypnea. Cyanosis. Apnea. Seizures. Cardiac arrest. Risk to preterm babies. Glycogen stores increase rapidly in the last month of the third trimester. Preterm infants are born before this occurs. So, the little amounts of glycogen available are used up rapidly and their supply is depleted. Table 10.9 Management of neonatal hypoglycemia. Prevention. Monitoring. Treatment. Increase awareness of conditions that predispose an infant to hypoglycemia. Early screening of at-risk infants. Early and frequent feedings. Maintain temperature. Complete evaluation and review of systems. Early breast or bottle feeding. Within 30 minutes. Glucose monitoring within 1 hour. Monitor pre-feeding levels. Thereafter. Oral feedings as tolerated. If glucose is very low or the infant is not able to feed orally. 2 ml/kg of 10% dextrose. Water as 4 bolus. Follow-up screenings within 30 minutes. Repeat bolus if glucose is 5 mg/dl. If unable to stabilize glucose level, consider increasing for rate or glucose concentration. Summary. Carbohydrate metabolism is the whole of the biochemical processes responsible for the metabolic formation, breakdown, and interconversion of carbohydrates in living organisms. Important pathways in carbohydrate metabolism include glycolysis, TCA cycle, hexose monophosphate, HMP, shunt, glycogenesis, glycogenolysis, hexose interconversions, gluconeogenesis, oxidative phosphorylation, and Cori cycle. Glycolysis is a catabolism of glucose. It is a cascade of enzymatically controlled reactions that convert glucose into 2-pyruvates, aerobic glycolysis, or lactate molecules, anaerobic glycolysis, and produces ADP along with NADH plus H+. Glycolysis is the major source of energy in certain tissues, example RBCs and skeletal muscles. Glycolysis can produce energy in skeletal muscles even in absence of oxygen. Pyruvate dehydrogenase complex deficiency is one of the most common neurodegenerative disorders associated with abnormal mitochondrial metabolism. The TCA cycle or the Krebs cycle is a series of redox reactions that generate the high-energy molecules NADH, FADH2, and ADP through the oxidation of acetyl-CoA. Total ADP generation by one TCA cycle equals 10. The regulation of the TCA cycle is largely determined by product inhibition and substrate availability. Fluorostate, arsenate, and malonate are inhibitors of the TCA cycle. TCA provides intermediates for the synthesis of non-essential amino acids. For example, many amino acids are synthesized starting with the transamination of alpha-ketoglutarate or oxalostate. Net ADP produced per molecule of glucose by complete oxidation, in aerobic condition, equals 32. HMP shunt is an alternate pathway of glucose oxidation. It involves the utilization of all forms of monosaccharides with the liberation of CO2 and many side products such as NADP plus H+. Liver glycogen is responsible to maintain blood glucose levels, particularly during the early stages of fasting. Muscle glycogen is regarded as a fuel reserve for the synthesis of ADP during muscle contraction. The main steps of glycogenesis are 1. Phosphorylation of glucose, 2. Formation of glucose 1-phosphate, 3. Formation of uridine diphosphate glucose, UDP glucose, 4. Formation of amylose chain and 5. Formation of the branch point. Glycogenolysis is the catabolism of glycogen during which the breakdown of glycogen into glucose units takes place. There are two main steps of glycogenolysis: 1. Shortening of the chain, and 2. Removal of the branch point. Glycogenesis and glycogenolysis are controlled by the enzyme glycogen synthase and glycogen phosphorylase. An elevated glucagon or epinephrine level increases glycogen degradation, whereas an elevated insulin results in increased glycogen synthesis. Type Ia, von Gierke's disease, is due to the deficiency of liver and kidney glucose 6-phosphatase. Metabolism of fructose is somewhat better than glucose in diabetes mellitus, as it is not affected by insulin or its deficiency. Essential fructosuria occurs due to a deficiency of the enzyme hepatic fructokinase. Hereditary fructose intolerance is caused by a deficiency of aldolase B. Galactitol, dulcitol, is a sugar alcohol, the reduction product of galactose. It is produced from galactose in a reaction catalyzed by aldose reductase. Synthesis of lactose takes place by the reaction between UDP galactose and glucose. It is catalyzed by the enzyme lactose synthase or UDP galactose glucose galactosyl transferase richly present in lactating breast. Classical galactosemia is due to a deficiency of the enzyme galactose 1-phosphate uridyl transferase. Sorbitol is produced in the body from glucose by the enzyme aldose reductase. Pathological changes associated with uncontrolled diabetes are due to the accumulation of sorbitol. Sorbitol can be used as a laxative when taken orally or as an enema. Gluconeogenesis is the formation of new glucose, i.e., the formation of glucose from non-carbohydrate sources. The important sources of new glucose in gluconeogenesis are pyruvate and lactate, muscle, RBC, amino acids, skeletal muscle, and glycerol, fat of adipose tissue. Gluconeogenesis supplies body cells with glucose after 4 to 6 hours of the last meal and becomes fully active as stores of glycogen are depleted and continuously add glucose to the blood. Hyperglycemia occurs when the blood glucose level rises more than 180 mg/dl. Hypoglycemia occurs when the blood glucose level decreases to less than 50 mg/dl. References. Exercise questions. For MCQ 1 to 5 see Part 1, and for MCQ 6 to 10 see Part 2 of this chapter. MCQ 11. Which of the following action of glucagon occurs in response to hypoglycemia to increase the blood sugar level in a normal healthy person? Glycogenesis, glycogenolysis, ketosis, lipogenesis, proteolysis. MCQ 12. A 35-year-old woman is taking an anti-diabetic drug for diabetes type 2. One of the effects of this drug is to increase glycolysis. Which of the following acids will produce excessively in this patient? Alpha-ketoglutarate, citrate, lactate, malate, oxalostate. MCQ 13. Which of the following substances is deficient in a young man to suppress the TCA cycle who is diagnosed with type 1 diabetes mellitus? Alpha-ketoglutarate, lactate, malate, oxalostate, succinate. MCQ 14. A 50-year-old man of normal body weight and a known case of diabetes mellitus type 2 was brought to the hospital semi-unconscious with complaints of sweating, drowsiness, confusion, and difficulty in speech. History revealed that he took some oral anti-diabetic agents without food. Which of the following would be the most likely cause of this condition of the patient? High blood glucose, high urine output, low blood glucose, low body temperature, low urine output. MCQ 15. An obese individual is brought to the ER. The patient has been on a starvation diet for 4 months and has lost 30 kilograms while consuming only water and vitamin pills. Which of the following would expect to be elevated in his blood? Acetate, acetoacetate, bicarbonate, bilirubin, glucose. MCQ 16. Which of the following metabolisms represent the synthesis of glucose from glucogenic amino acids, glycerol, and lactate? Gluconeogenesis, glycogenesis, glycogenolysis, glycolysis. HMP shunt. Please write answers in the comments or contact drmjot@yahoo.com. End of Part 3 of 3 of Chapter 10. Thank you.